Speaker is Tyler Dross, and he will be speaking about tissue culture of Virginia Bluebells, and his mentor is David Foster. So Tyler.
Alrighty, so my goal today is to give you guys both an introduction into the world that I have lived in for the past year and a half now, but also to get you a little excited about what I do and about Bluebells. They're an absolutely gorgeous plant, but there are also absolutely little buggers, and it's gonna be a wild ride. So stay with me. Virginia Bluebells, also known as Mertensia virginica, are a perennial in the Boraginaceae family. They're native to eastern North America, including Pennsylvania, and they are widely used as an exterior home decoration. Classically, we seed and tuber propagate them. This is a slower process, and this is one of those things that when we're looking at this from a commercial standpoint, we want to speed up this process. We want to make sure that it is at its most efficient. Classically, this is a picture I took at Shanks Ferry Wildflower Preserve. They look like that. They're these blue trumpet-shaped flowers in kind of a cone. But we, on campus, in our beds, have a white variety of them. And one of the main reasons for doing my research is because of this white variety. If you were to seed or tuber propagate this, your chances of getting the white variety are significantly lower than getting the blue variety, even if you start with the white variety as your base. It's just how they happen to work out. So in tissue culture, we can guarantee the white variety if we can ever get it to be successful, which is the entire purpose of me doing my research. This has been a project for over a decade. It took more than 10 people so far, and Dr. Foster spent an entire sabbatical on it. So why I took this is because I like a challenge, and Bluebells are a really fun challenge. So we'll see. My research questions, just to make it incredibly clear, are what variables are preventing Virginia Bluebells from being propagated in tissue culture? We have three that we think might be a part of it: etiolation, which I'll explain in a little bit, flowering, and age of the tissue. And how can we overcome these variables in bluebells and other related species? Like I said, the white variegation is our really big key thing here, why we want to do it. But also trilliums are a related species, and trilliums are ridiculously expensive if you've ever tried to buy them for your garden or outdoor decorating. If we could figure that out, we could lower that price significantly. So there is an economic benefit to this research. I wanted to give you guys some photos here, just so you know, you can see things the way I see them. When I'm saying certain terminology, it'll make more sense, because otherwise these are just a bunch of words that you may or may not know what they mean. Etiolated tissue is tissue that's grown in the dark without the presence of sunlight. In our lab, we grow them in essentially a fridge. It's called a Percival environmental chamber. And that way, you don't get access to like chlorophyll and those kind of things in the plant. Phenolic activity, or I'm going to refer to it as browning, is just a byproduct of plant metabolism. When plants are active and they are growing, they produce phenolic activity a lot of the times depending on the hormone. Not every hormone, but it depends. In our hormones, they do. Roots and shoots. If you have taken any of our entry-level biology courses, you probably know this, but these rooting are these fine hairs, and the shoots are the main stalk of the plant. And then callus tissue is just undifferentiated tissue, meaning it is tissue that hasn't decided what it wants to do yet. I always describe it as plant cancer, but don't think of it in that negative sense, but it's a tumor almost. It's callus tissue. And those are those little globules on top of the guy that looks like a grasshopper. So, what was our source tissue? We used three different plant groups. We used young plants, which will be classified for our purposes 0 to 1 years old. Middle plants, 1 to 2 years old. The middle category is really important because they still haven't developed their flower buds. They don't develop flower buds until year 3. And those flowers are pre-formed. So once you finish a season, you've got the flower buds for the next season. And then old plants are anything with a flower bud, usually 3+ years old. We had 2 different treatment groups. It was either stratified in the dark at 4 degrees Celsius for 2 to 4 months, so we grew them in the fridge, or we took them straight from the outdoor beds for Dr. Foster's home garden. We used 4 different hormones in our research at various different combinations. Thidiaziron, TDZ, is a synthetic cytokinin. When you think of cytokinins, I want you to think shoots. Cytokinins produce shoots. And then we used 3 different synthetic auxins. We used NAA, 2,4-D, and IBA. I'm not going to even try to pronounce those because that wouldn't be fun for any of us. When you're growing in tissue culture, your media is key. You're going to provide all the nutrients the plants need. You're going to provide a base for what the plants grow on. But ultimately, the thing that's going to be variable is your hormones that you're using. And those things produce the shoots or the roots, right? So cytokinins produce shoot growth. Remember C for shoot, and then auxins produce root growth. And ultimately, my job is to find the specifics of those ratios and try to keep things sterile. And that's a lot harder than you would imagine. So our media is an agar media with myriskyne and Scootage Plus. It's got novel hormone concentrations. I'm not going to read all those. You can read those, but it's just a bunch of different combinations of auxins and cytokinins. And our goal is just to get shoots and roots on the same plant. We're trying to take cut plants that we snip and grow roots on them, or we're trying to take the same plants, grow roots, and then produce new shoots in case the old shoots die. So the ultimate goal is to create a bunch of little bluebell plants from one identical plant. So you're creating a bunch of various tissue from a single source. In tissue culture, there's 2 different methods, like I just said. You can either de-differentiate the cells, making them into this callus tissue, and then put roots on them, and then you could have something called a somatic embryo, which is just like a baby embryo. It's like a baby plant. And you're gonna get the sprouts from those somatic embryos to form the shoots. Or you can take a live cutting, and as long as it stays alive and you get roots on it, you had a success, right? The second method would be ideal. It's just a lot more difficult than we imagined, and there's a lot more issues with sterility in the second method. But we do try both. Here's an image of what it looks like over the course of about 4 weeks. You have a lot of phenolic activity by about 4 weeks in, and phenolic activity is really just a bugger when it comes to our research. It's one of those things that we try to keep as low as possible because it interferes with metabolism. But you can see the cuttings put into the media. You just keep going, and if you look close enough, you can see swelling on the day 28s in the same plants. It's really cool. But the swelling usually refers to the callus tissue forming, and that is something we want, because remember, if we can de-differentiate things into callus tissue, that's the first step for us. In the IBA media, this was just using IBA, we had a huge success. We were really excited about this because we got callus tissue. The browning was delayed significantly. I left it for over a month, and these are pictures after a month, not after 4 weeks, but after a month, 2 months, I left it over the break. And we had roots, we had callus tissue all forming on the same plants. We didn't have live shoots yet, and we did not get somatic embryos from this, but this was the first step, and this was critical. Some of these are under the microscope just so you can see the fine root hairs that form. These things don't like to be disturbed, and so that's one of our other issues, is if we were to try to transplant this to another media, we'd likely fail and it'd likely die off because they're just persnickety little plants. In IBA and TDZ combination media, this was kind of testing a new hormone that we hadn't used in our lab before. TDZ had shown hope in other plants, and so we wanted to try it on bluebells. But what this ultimately did, as indicated by the arrow, is showed what age group of plants is going to be most likely to be used in our research, and what is most viable to be used in our research. Browning intensity was sort of high across all of them. It wasn't something that we could really control, and we think it's to do with the TDZ media itself. TDZ is just, it doesn't seem to be the right hormone for us. But if you look at the callus tissue, and what the arrow is pointing at is that yellow bar being high for us, we had a lot of callus tissue growth in the middle group, that 1 to 2 year old plants, meaning that that is probably what we're going to use from here on out. Older plants, but they still don't have preformed flower buds. When talking about outdoor versus indoor beds, we use 3 different combinations of media: IBA, NAA, and TDZ by itself. And if you look here, you're going to get the most callus tissue with NAA grown in an outdoor bed. So we had seen this before. We have had one success in our lab where a root and shoot were on the same plant, and that was in 2 ppm, 2 parts per million NAA media. And so this doesn't shock us, But it does further confirm that we're on the right track. We need to be working with this media. This is the media that's going to give us the best results. On the etiolated tissue, you're going to see that those callus tissue levels are a lot lower on this graph. If you look between the two, you've got a lot lower on etiolated tissue. So it wasn't our magic bullet that we were expecting it to be. So, like I said, etiolation wasn't the magic bullet that we expected it to be. We were hoping that if we grew them indoors and stratified them in the cold, that was just going to be the switch. There was something about cold tolerance or something about the light tolerance that it was going to be the thing that let us grow bluebells. It wasn't, sadly. So we've decided that we need to get these shoots before the roots, right? So we're going to keep working on taking the cuttings and trying to root the cuttings, or producing somatic embryos before we produce roots. We did decide that our ideal tissue is about 1 to 2 years old, right before its preformed flower buds, but still old enough that the plant has developed a little bit and it's not in like an embryonic stage. We don't quite want to make any big determinations here, but we think outdoor tissue is going to be a better candidate than indoor tissue grown indoors, based on these graphs here and the results we had from those. The biggest problem with that is sterility. A lot of the times we're just going to get a bunch of bacteria that are growing in the tissue culture tubes, and those interfere with the growth of the plant, and so that's a negative thing. And if the bacteria is just taking over our tubes, then we're not going to be able to do anything with those plants, right? They're going to be infected for a long time. It gets in the soil. It's really annoying. Anything about tissue contamination is just frustrating. So our next steps: more hormones, more sterile conditions. We want to sterilize we want to re-sterilize both the Percival chamber and the potting media. We want to try sterilizing the potting media before we use it in the etiolated tissue. And ultimately, we just keep trying. This is one of those things that we don't want to give up on, because bluebells are such a really cool plant, and so just wonderful. I have really grown to love these little things, even though they have given me a lot of pain, but I see why Dr. Foster has spent so long on this, and so I want to acknowledge Dr. Foster for being my professor, my mentor, and my friend. Messiah University Department of Biological Sciences for the funding and lab space, and Dr. Bogdan Barchadwal for presentation guidance on this. I want to open up the floor to any questions.
So, Tyler, as somebody who's interested in a career in medicine, why are you involved in what benefits are there?
Well, I could give you the answer I give everyone, or I could give you the real answer. So the real answer is there's a lot of fine details. It's all the fine motor skills. You know, Dr. Foster will tell you his most successful tissue culture student went on to become a surgeon. So these fine motor details that I'm doing under a hood in sterile conditions are going to cross over into medicine. But also, these plants are just so dang cool. I mean, they're just something I'm fascinated in. Dr. Foster got me fascinated in them. And they're something that it'll carry with me. I think we had a question over Zoom.
Yeah, are we allowed to ask questions if we're just part of the Zoom group?
Yeah, go for it.
Okay, so why, you mentioned that outdoor grown tissue may prove most successful. Do you have any just hypotheses or even just, wild guesses as to why that might be the case?
Sure, so when you, oh, go ahead.
Just from the standpoint of mechanism?
Yeah, so when you look at the tissue, you want to mimic their natural environments as much as possible. And so it would make sense that an outdoor grown tissue would do better than something grown in my lab in a fridge in the dark. It is mimicking those conditions. Again, the biggest problem with those, though, is that they are so easily contaminated by various bacteria. Hyphae is a real bugger in our lab for microbiologists in here. So, yeah. Other questions? Yeah.
You mentioned that there was like research of this about for 10 years previous to this one. Can you talk more about that?
Sure. So over the last, Dr. Foster, 10, 15 years now?
Yeah, pretty close.
so many different hormones have been used, so many different sterile techniques. They tried bleaching the tissue until it practically died. You know, if I had been earlier, I would have been doing tests rather on how long can this tissue survive with bleach. Now it's 3 minutes and we just sort of know it. We've used so many different hormones over the years, so many different types of tissue. They've done seeds before. Yeah, it's been crazy. Dr. Foster.
Yeah, I think I'll just point out the significance of Tyler kind of figuring out what age class of plants is responding the best. We really think there's a genetic block in being vegetatively propagatable caused by that preformed flower bloom. It's pretty significant in a lot of plants. Once they're in flower, they're no good for vegetative propagation. And as Tyler mentioned, that's also common in Trillium. Nobody has been yet able to tissue culture Trilliums, and a lot of people have tried. So
And how much is a Trillium bulb?
$25.
Yeah.
But
So
once the goal is to get down to a plant that's repeatable, then to go into genetically what genes are turned on, what's allowing it.
yeah.
What was your most exciting result for?
I think over the winter break, coming back and seeing these guys, I was so excited because when we got rooting, that was consistent across almost every single one of my samples, and I only had one of my samples contaminated. The biggest problem with contamination is that it gets in between the two cell wall layers, and so when you cut it, you release it, right? And we were getting that across everything, so it's a crapshoot when it comes to if it's going to be contaminated or not. It's not a technique thing at this point. If you had come to my presentation last semester, it was all about technique, all about sterile technique, but it's not a technique issue anymore. It was just, we got really lucky with these IBA media plants. Any other questions?
So Tyler, what's the next step after, if you get like the roots and shoots, what would you do with those then?
Yeah.
To getting them to the plant stage?
So we're gonna freeze them at -80, and we're gonna go to our friends at Penn State Harrisburg. Dr. Syram is there, and we would be working on looking at the genetic element, seeing what that gene is that's turned on by the flowering, and seeing if we can just figure that out. It becomes plant genetics at that point.
Mm-hmm.
So it would be fascinating.
Mm-hmm.
Thank you, guys.